Good afternoon, all of you. Welcome for the biology classes. Dear students, we have studied the chapter 5 already completed. In chapter 6, molecular basis of inheritance, we have studied many subunits, say nucleic acids, structure, component, DNA and RNA, and the structure of DNA, Watson and Crick model of DNA, the experiments, DNA as genetic material, Griffith experiment, every experiment, Hershey and Chase experiment, semi-conservative replication of DNA, how the DNA replication takes place, what are the enzymes helping for this replication of DNA, and also there is some experiments and packing of DNA, and also central dogma of molecular biology. What is the central dogma of molecular biology? DNA, messenger RNA, mRNA, and protein synthesis. Our cell or DNA produces proteins. Cell produces proteins according to the message present in DNA molecules. It requires some component. RNA is required for protein synthesis. All the types of RNA required for the process of formation of polypeptide protein synthesis. The central dogma of molecular biology, you know that the transcription. What is meant by transcription? The copying of one strand of DNA into RNA is called as transcription. The transcription, you know that DNA is genetic material. The properties of DNA. DNA is a very stable molecule. It has a double-stranded, while RNA is the single-stranded. In this period, we will discuss about, already discussed about transcriptional unit. In this, the transcription unit and gene. Transcription unit and gene. What is meant by the genes? The gene is the functional unit of inheritance. The gene is the functional unit of inheritance. The gene is the unit of inheritance. It is made up of segment of DNA. Gene is the unit of inheritance. Functional unit of inheritance. It is the segment of DNA. In the DNA, there will be out of genes are present. They carry the characters from one generation to another generation by replication of DNA, meiosis. Therefore, the gene is the unit of inheritance, functional unit of inheritance. Gene is is functional unit of inheritance. Functional unit of inheritance. Gene is functional unit of inheritance. It is the made up of segment of DNA involved in producing a polypeptide. It is the segment of DNA. Here is the DNA. The genes are present. One part of DNA. Gene is the segment of DNA. It is a segment of DNA to form made up of involved in process of producing polypeptides. It is a segment of DNA. Gene is the segment of DNA for the process of producing polypeptide, for the production of polypeptide or protein. Another name is polypeptide or protein. Gene is a functional unit of inheritance. Okay, it is the segment of DNA for the production of polypeptide or protein. This polypeptide chain oil to form. This polypeptide chain, you know that the protein is, there will be primary, secondary, tertiary and quaternary type of proteins. The proteins are in the ball-like or coiling-like structure. Therefore, this protein or polypeptide chain oil to form protein molecule. This polypeptide or protein polypeptide coil to form this polypeptide chain called to form polypeptide chain. This polypeptide chain called to form protein molecule, a large chain of molecules. This polypeptide chain called to form protein molecules. This polypeptide chain called to form protein molecules. This in which it controls the different or an enzyme which in turn controls different traits or enzyme protein molecules or enzyme which in turn control characters. Therefore, the DNA containing the gene in the coded form. You know that the coded form is the nitrogen bases AGTC, AGUC in RNA AGTC formation. Therefore, there will be two strands of DNA. It will become AGTC. The complementary codon or nitrogen bases is ATGCTACGATCTAG. This is the complementary on another strand. This is the template strand. Is coding strand in the transcriptional unit. Therefore, the gene is the functional unit of inheritance. and it is a segment of DNA for the production of polypeptide or protein. This polypeptide chain oil to form protein molecules or enzyme which in turn control characters. Enzymes or protein govern the controls the character. Therefore, the gene is the unit of inheritance. The fine structure of gene. The fine structure of gene according to Simon Benzer, the gene is defined as the unit of function called cistron. The gene is the unit of function is called as the cistron. This is about gene, the fine structure of gene. According to the gene fine structure or ultra structure, detailed structure. According to Simon Benzer, the gene is the functional unit is called as cistron. Cistron, according to Simon Benzer, gene is a unit of function called cistron. Gene is a unit of function. Unit of function called as cistron. The gene act the function made some duties. It is called as cistron. Okay, the gene is a unit of function called as cistron. This cistron again, the cistron also known as structural gene, segment of DNA coding for polypeptide. This cistron is the segment of DNA that coding for polypeptide or protein. Particular protein will be coded by according to this cistron. Genes present, a group of genes present on DNA template molecules DNA. Therefore, it is a unit of function called cistron. Unit of function is called cistron. It is also known as structural gene. Structural gene. Cistron is also known as structural gene. It is a unit of function called a cistron. This cistron undergoes some changes. There will be record. Each cistron undergoes the segment is called as racon. Each cistron has several racons and newtons. Each cistrons has several racons and newtons. Each cistron has several racons and newtons. Each cistron has several racons and newtons. What is meant by racons? Racons means it undergoes mutation. It undergoes recombination. What is meant by recombination. The formation of new gene recombination during crossing over in meiosis is called as recombination. You may remember in first year the meiosis, how the meiosis takes place, what are the important stages in meiosis: leptotin, zygotin, pectin, diplotin, and dikinesis. During this phase, the chromosomes undergoes the shuffling of characters. The part of chromatic, sister chromatids cut down, and exchange of these parts takes place. This is about linkage and crossing over takes place. Here is the recombination. The unit it undergoes is called as recombination. Cistern undergoes recons, and unit of recombination is called as recons, and it also this recons undergoes mutation. You know that a sudden change in the genes is called as mutation. Due to the mutation, there will be some problems arises in the human living beings genes. It is called as disorder. You know that genetic disorder, hemophilia, color blindness, etc. is the genetic or Mendelian disorder. Here is the G is the unit of function is called cistern. Cisterns again contains many units because it undergoes recombination. The unit of recombination is called as recons. Unit of recombination. Recombination is nothing but the formation of new genes during crossing over in meiosis is called as recombination. It undergoes mutation. Therefore, the unit of mutation is called as mutons. Therefore, these cistron, the fine structure of the genes, the DNA, this is the segment of DNA, is called as cistron. Cistron again, this segment, this cistron will undergoes some parts. It is called as recons. This recons undergoes another split is called as muton. So, in this, there will be the transcription takes place. Translation. The mRNA copies this cistron part, this segment of DNA copied by mRNA. This is called as transcription, and again it will be translation, and protein will be formed. Protein molecules will be formed. It is in the coiled nature. You know that the different types of proteins are present: primary and secondary, tertiary, and quaternary proteins. Hemoglobin is one of the proteins. It contains 2 alpha and 2 beta chain. This is the fine structure of translation. And in this is a prokaryotic messenger RNA, clear. But in eukaryotic, there will be because of it is present in nucleus. Therefore, in prokaryotes, there will be a definite nucleus. There is absence of definite nucleus, Therefore, it will be present in the cytoplasm in eukaryote. There will be a definite nucleus is there. Therefore, this heterogeneous RNA, mRNA undergoes some processing. It is called as complexity 1 and complexity 2. RNA polymerase containing or transcribes all these three types of RNAs: transfer RNA, messenger RNA, and heterogeneous RNA and ribosomal RNA. Therefore, in eukaryote, there will be the coding exon and non-coding introns. In eukaryotes, there will be a hnRNA means precursor of mRNA. Before it will be a processing, after copying, processing and becomes mRNA. It becomes pre-mRNA. Therefore, it is precursor. It contains exons and introns. The exons will be interrupted by intron. This is the exons. Exon is the coding sequences. And intron is the non-coding sequences. During the processing is called as splicing. Introns will be removed and exons will be joined. Introns will be removed and exons will be joined. So the continuous process will be for continuous. There will be a continuous process. It is called as messenger RNA, as in prokaryotes. Therefore, this transcription unit and gene, the functional unit of inheritance and file structure according to Seymour-Bengin, gene is a unit of function called cistron. The unit of these systems has several recons. The unit of recombination is called as recons. The unit of mutation is called mutons. Therefore, here about the transcription unit and gene. Next, we will go to how the transcription takes place in prokaryotes. There will be a schematic representation. How the transcription takes place. Transcription is nothing but the copying of one strand of DNA into mRNA. mRNA is very, very important. You know that the DNA having the two strands. This is the template strand, and another one is the coding strand. The mRNA copying. You know that you are a student. You copy the notes, important book, or in the classroom. You have in the pen, pencil, rubber, and notebook. You have copied fast during the lecture. Copied fast or write it in the short form. Very short form, biology, male and female, etc. Very, very short forms there. Likewise, the mRNA comes near the template strand and copy according to the complementary rules. What is the complementary rule? See the 5 3 and 5 and complementary is 5 and 3 according to the pentose sugar. The position 1, 2, 3, 4, 5, fifth position pentose sugar, and here is always one will be remembered, the catalyzation or transcription is in the only one direction, 5 raised to 3 direction. Even in RNA or DNA, always there will be a direction 5 dash to 3 dash direction, 5 prime to 3 dash direction. We read the book in left to right, not maximum people's left to right. Reading and writing is left to right. Likewise, there will be the transcription. The transcription means the copying of information present in the one of the strand of DNA. One strand of DNA. Here is the information AGTC, AGTC, AGTC. The complementary one is A is T, G, C, A, T is A. Likewise, that will be present complementary because of two polynucleotide strands antiparallel and complementary to each other. And in this this information you are copied the important notes in the classroom only. There will be no textbook and any other materials. The while lecturing to the students, it will be copied. So you attend the classroom. Therefore, the mRNA lies near to the template strand DNA and copy the matter, but it is in the direction of 5 dash to 3 dash direction. Here is the left to right. Therefore, 5 dash to 3 dash. It contains A. A is converted into T. There will be no T in the mRNA. T is replaced by U. Know that it is U. G is C and G is C. T is the A. T is A and C is G. A again U. This is a copying in the complementary base pairing. Here the RNA contains UCA or GU say here C. This is the one codon. It controls one proteins or many proteins. In this way, it is called as transcription. Transcribe. Note down the information present in this line. What are the information present in the DNA molecules? Therefore, it is called as transcription. How transcription takes place? You know that transcriptional unit contains the structural gene and promoter and terminator, P and T structural gene. This is the template strand, is the coding strand. You know that template strand, coding strand, and the direction is also 3 dash, 5 dash, 5 dash, 3 dash. Always in this direction, 5 dash to 3 dash direction. This is called as upstream. This is the downstream. Promoter is present at the 3 dash end of template strand or upstream, and vice versa. This is the structure of this transcription unit. And again, what is the steps in the transcription in prokaryotes? Transcription Transcription in prokaryotes. Transcription in prokaryotes. Transcription in prokaryotes. Here there will be a schematic representation. Schematic representation. The DNA is the double stranded structure and highly coiled structure. DNA is the highly coiled structure. Here is the terminator. Promoter, and yeah, initiation takes place in the transcription unit. There will at the promoter end there will be a core enzyme is present. In this there will be a sigma or initiation factor will be present. The RNA polymerase is very important in this. All the three types of RNA are needed to synthesize a protein in a cell. All the three types of RNA. There is messenger RNA and also transfer RNA and ribosomal RNA is required to form the protein. There is single DNA dependent RNA polymerase that catalyzes the transcription of all types of RNA. Catalyzation, formation of this RNA will be needed. Therefore, the RNA will be formed. This RNA transcribe, read the information, write it, and after the completion of these transcription or writing word, it fall on the cell and for another functions or making the another function. This is the promoter and again there will be one of the diagrams. Open loop will be formed as these are promoter and again here is the sigma bond is very important. There will be a RNA polymerase and attached with the sigma bond. This is the RNA polymerase enzyme required for transcription. The row, this is the sigma bond of factor. It starts just like starter in tube light. It is located at the promoter end and starts the transcription. Starting the write down work. Therefore, RNA polymerase attached to the promoter and sigma gives the start signal for this copying of the information present in the DNA. And again, it moves along. This is the process is called as initiation. The process is called as initiation as these sigma factor moves along this RNA, it forms the short stretch of RNA, mRNA. This mRNA copies down all the information present. As the mRNA goes to the middle line, and by the help of another factor, row factor, the loop formation is formed. As the loop formation is formed means it will be transcribed definitely, correctly transcription takes place. Along these loop open and close takes place. Here is the mRNA, here is the loop formation, sigma bond, loop formation formed. Along these, lastly, this mRNA falls on the cell cytoplasm. After the completion, this is the terminator. Initiation, the elongation takes place. Elongation requires some energy. Triphosphate nucleotide triphosphate elongation takes place. As this moves along this chain, it copies all the information present, and mRNA copied all the information and falls in the cell. This is last. It ends while the signal for termination until the signals will be formed by these row factor. This is the row factor, and RNA polymerase. Enzyme is very important. RNA polymerase enzyme is very important. Elongation and termination. Elongation and termination. Here is the 5' and 3' and 5' be taken. All these in the form of 3' and 5' 3' and 5' and 3' 5' 3' totally 3 steps will be formed in this bacterial transcription or prokaryotic transcription. Okay, this is the promoter and this is the terminator, and in between there will be a step is called as elongation. By some energy is required for the loop formation, the stretching of this or breaking the bond. The RNA polymerase requires some energy, and after during the loop formation, all the information will be copied along this length of DNA. Where the genes is present, the genes is in the coded form. Lastly, it ends while the signal for termination. Therefore, 3 steps are very important. Here is the function of sigma is the initiation factor, rho is the end factor, termination factor. During this, there will be all the information present in the template strand will be copied. It is called as transcribed. Transcribed mRNA is in the form of this. This is the transcribed mRNA in prokaryotes. But in eukaryotes, there will be some process will be required. It is called as complexities. complexity is 1 and complexity is 2. All the three mRNA transcribe all the three types of RNA. RNA polymerase here transcribes the all types of RNA. In eukaryotic transcription, there will be additional changes will be required. It is called as complexities or post-transcriptional changes. Complexities or post-transcriptional changes. Okay, complexities 1 is the RNA polymerase 1 transcribe some RNA, tRNA and heterogeneous nuclear RNA, mRNA, heterogeneous nuclear mRNA. RNA polymerase 1, RNA polymerase 1, 2 and 3. RNA polymerase 1, 2 and 3. It transcribes, what is the RNA? It transcribes the additional complexity transcribes rRNA. It is very important. Ribosomal RNA. It transcribes the ribosomal RNA. It is very important in transcription and translation. Ribosomal RNA. This RNA having the site 28S, 18S, 28S, 18S and 5.8S. Set by unit 5.8S. And second, RNA polymerase 2 is the hnRNA or precursor of RNA. hnRNA or precursor mRNA. Before formation of mRNA, it will undergo processing because this is the heterogeneous RNA. HN means heterogeneous nuclear RNA. Because it is present in eukaryote, there will be a definite nucleus is present. This RNA present in the nucleus. And third one is the transfer RNA. Transfer RNA also contains some transcribe 5S, 5 sRNA, snRNA, 5S RNA and snRNA, small nuclear RNA and SR small ribosomal RNA, small 5S or small ribosomal small nuclear RNA. It transcribes the tRNA. Therefore, RNA polymerase, it is very, very important. It is the eukaryotic transcription is division of labor, justify. Just you give the function of these RNA types of RNA polymerase and function. Function is it transcribes the RNA. RNA polymerase transcribe the RNA, ribosomal RNA, hnRNA and transfer RNA. These three will be very, very important in transcription in hnRNA. will be additional complexities. It will be pre-processing takes place before it undergoes the transcription will be processing. Therefore, the complexities justify eukaryotic transcription is is a division of labor. Question: eukaryotic transcription is division of labor. Justify. This is the answer. RNA polymerase 1, 2, 3. What is the function? It transcribes the RNA, ribosomal RNA, and hnRNA and transfer RNA. This is the answer. And second complexity is first is this. Second complexities is it undergoes splicing, capping, and poly A because it is interrupted by introns. You know that this complexities 5' to 3' direction, it has the coding exons and non-coding introns. Coding exons and non-coding introns. This is the exons. During the processing or splicing means the removal of introns because it is non-coding part of this mRNA. It will be removed and joining of exon takes place. And second is the adding of 5'3" direction. There will be a capping. This is the splicing takes place. Here is the capping. Capping means Mg methyl gonotriphosphate. The adding at the 5' and methyl triphosphate guanine plus. This is called as capping. Here is the 3' and lastly this will be Mg 3pp. Lastly the 3' and poly A poly about 200 base of adenine is added, poly A tail. After this there will be a mRNA is formed. This is the splicing take place. Second complexities is the splicing, capping, and polyadenylation. Polyadenylation because number of these adenine will be added at the 3' and is called as poly A tail. It is called as poly A tail. This is about how the transcription takes place. You know that this transcription is the copying of one strand of DNA into mRNA. mRNA is very, very important for copying the template strand, the information present in this is the information present in the template strand. According to complementary base pair, the RNA contains U instead of T. There is no T in the mRNA because the ribosomal RNA it is copied. This copying is called as transcription. The copied this strand is called after you are copied all the notes in the class. It is the copy or note down points. All the points will be noted down or copied. Copied is called transcribed mRNA. Transcription takes place. Therefore, in eukaryotes, there will be prokaryotes. These steps is there: initiation and elongation and termination. Initiation is the rho factor, elongation is the sigma factor, initiation sigma factor, elongation is the rho factor, and also termination is the rho factor. And in eukaryotics, there will be a complexities. There are two complexities. One is the complexity 1 is the transcribe 3 RNA polymerase 1, 2, 3 transcribe the rRNA, hRNA, and T transfer RNA. And second complexity it undergoes heterogeneous RNA undergoes splicing. Splicing means once again removal of introns and adding of coding sequence exons, and in additional is capping and poly A. This will be the structure. The common thing is the 5 prime and 3 prime and double stranded. Here is the exon and intron. What is about the gene, fine structure of genes. In examination, describe the transcription in bacteria or prokaryotic transcription. Schematic representation, 3 points will be should be written, and also second, eukaryotic transcription. What is meant by splicing, capping, poly A. Splicing is nothing but removal of introns and adding of joining of exons, and capping is the MG triple phi. M means methyl guanosine triphosphate is added to the 5' end of this mRNA, heterogeneous nuclear RNA, hnRNA, and lastly the poly A tail will be formed. Added a number of these up to 200 adenine will be added. Therefore, polyadenine tail. Polyadenylation is called a polyadenylation means the adding of many adenosine at 3' and it is very, very important. It is called a polyadenylation. Okay, this will complete the process of transcription. In next class we will discuss about the translation. Thank you.